So we started looking at cancers and mutations, and basically we did this quick overview of the 6 main points of how cancers are different to cancerous cells, tumour cells are different to normal cells. And these were the 6 main differences. So if we had a look here, that was a tumour and they were our normal cells. So we basically said the key main identifying features of a cancerous cell a tumour cell compared to a normal cell would be that they have irregular shape. Their nucleus is actually darker than a normal cells and larger, and sometimes the cells may even have more than one. In tumour cells, not all proteins that are needed are produced, so they can actually work differently. They'll have different antigens on their surface. They don't respond to growth regulation processes, and they have that more frequent cell division. So that was a really super quick overview of how tumour cells differ to normal cells. And we basically just talked about cancers. Obviously, we have benign and malignant. And if we just do a quick summary here of benign and malignant, benign, and we'll just summarize them really nice and quickly here. So what are the differences? Well, benign, the main difference between benign and malignant is a benign tumour is not cancerous. That is our main initial difference, whereas malignant ones we class as being actually a cancer. So that's our main difference. Benign grows slower than malignant. So by growing slower, I'm comparing it to the malignant, and our malignant grow rapidly, and they also invade and destroy surrounding tissue. Benign very rarely invade and destroy surrounding tissue, and that is because they're often covered in fibrous tissue. So they're often covered in fibrous tissue. And that stops them invading others. That stops that invasion. I'm going to just put other there. Stops invading other tissue. They are harmless, but they can cause blockages. So I'm just going to put can cause blockages. And put pressure on other organs. Some have potential to become malignant, not all. So some can become malignant. Yeah, malignant. I just had a moment then spelling it. So malignant, we know they are cancer. They grow rapidly. They invade and destroy surrounding tissues. The cells can break off. And when they break off, they can then travel to other parts of the body, and they can travel to other parts of the body either in the bloodstream or in the lymphatic system. We also looked at 2 different things called tumour suppressor genes and proto-oncogenes. Tumour suppressor genes are genes that suppress tumours from forming, and basically when they function normally, these tumour suppressor genes slow cell division, whereas a proto-oncogene stimulates cell division. I don't think I've got a bit like that. And then we had a quick discussion about estrogen and tumour cells and methylation. So I'm just going to summarise. Yeah, I'm going to quickly summarise tumour suppressor proto-oncogenes as well. So let's just pop them here. So yeah, tumour suppressor genes. A quick summary: tumor suppressor genes. These can be inactivated. So I'm just going to put can be inactivated, can inactivate if a mutation occurs. How do they work? Well, when functioning normally, they prevent, or they slow, it slows cell division. And it does this by producing proteins that stop cells dividing and causing them to self-destruct. Whereas our proto-oncogene when they become mutated, we obviously call them an oncogene. Get rid of the proto in front of it. And if they're working normally, when they're working normally, they stimulate cell division. We obviously want cell division to happen, but if these get overexpressed, they can increase the rate of cell division, which can obviously be really bad for your body. So yeah, let's have a quick look at some of these exam questions. I think would be a sensible thing to look at now. Share. Here we go. So this is our first question, and it's all about estrogen. So it's why I haven't gone into loads of detail about estrogen now, because it's going to go over it in the question. It's quite a long question, and there's a lot of information here about drugs trials and how they work, and it actually links to lots of different parts. So we're going to look at this in quite a bit of detail. So I'm going to read it out, and I'm going to highlight parts and sort of pick out for you which bits would be really important for us to pay attention to when answering these sorts of questions. So if I find my pen, here we go. So estrogen is a substance produced by the enzyme aromatase. So straight away, I would highlight the hormone estrogen. We know it's going to play a key part in this question, so highlight that. Okay, so we're talking about estrogen. Substance, it's produced by the enzyme aromatase, so I'd probably highlight that as well. In females, the main source of estrogen is in the ovaries, but aromatase is produced by many other organs in the body, including the lungs. Estrogen can stimulate the development of some lung tumours. That's quite interesting, so I'm going to highlight that. So it can stimulate the development of some lung tumours. And in these tumours, binding of estrogen to cell surface receptors stimulates cell division. So then it goes on to explain the experiment. So scientists investigated whether 2 drugs could prevent lung tumours in female mice. First, what they did is they removed the ovaries from these mice. Then they injected the mice with a tumour-causing chemical found in tobacco twice a day for 4 weeks. The mice were then randomly allocated to one of the 4 groups, and each group contained 10 mice. And then it goes through to explain the 4 different groups. So we had group Q, and they were given the placebo. So I'd highlight that straight away, that we're talking about placebo here. And in this situation, placebo did not contain either drug. Group R was given anastrozole, and this inhibits the enzyme aromatase. S was given fulvestrant. This binds to estrogen receptors. And group T was given both drugs. So that's really important to note as well. So the mice were given these drugs each week during weeks 5 to 15 of the investigation. The scientists removed the ovaries from the mice for the investigation, and they also gave the mice injections of the substrate of aromatase each day. Explain why these steps are necessary. So these are the steps I'm about. Scientists removed the ovaries from the mice, and they also gave the mice injections of the substrate aromatase each day. So 2 marks for this part of the question. It basically wants us to say why we're getting rid of the ovaries and why we're injecting the mice with aromatase each day. So first of all, well, the question is looking at how estrogen's made. And up here, we've been told in females, the main source of estrogen is the ovaries, but it's also produced in other parts of the body, including the lungs. Estrogen can stimulate the development of some lung tumours. And the scientists are investigating whether 2 drugs can prevent lung tumours in female mice. So in this situation, we're wanting to remove a source of estrogen. We're wanting to control this as much as possible, because some mice produce different amounts of estrogen to others if they're producing it naturally from their ovaries. So we're not going to remove we can't remove the lungs because the mice can't live without the lungs, and we're looking if the lungs are going to develop tumours. But can we remove the main source of where estrogen is being made in the body? Yes, we can remove the ovaries from the mice and it doesn't affect them. And because one mouse could produce a lot higher levels of estrogen than the other. So basically what that's going to do is that's going to get rid of variability in this situation. So how you would answer that is the ovaries are removed. Oh, that's a bit thick. Oops. So ovaries are removed to remove main source of estrogen. To remove the main source of estrogen, as different mice would produce different levels. Oh, as different mice would produce different levels. So that would get you 1 mark. You've answered that first part, 1 mark. Now we need to say why we're giving the mice the injections of a substrate of aromatase each day. Well, that's so that we can control that amount of estrogen. So then that second part of the question, so the injections are going to allow the scientists to control the level of estrogen. And then that would give you the 2nd mark. So the question then goes on to say, the scientists predicted that fulvestrant would be more effective. So here we go, that's the drug that's going to be more effective when given with anastrozole than when given alone. So use the information provided to suggest why they predict this. So let's go back and look at what this question is actually all about. So this question is asking us, they're investigating whether 2 drugs could prevent tumours in female mice. So we're looking at ways to prevent tumours. Anastrozole inhibits the enzyme aromatase. So we know estrogen is produced by aromatase. The estrogen can stimulate the development of some lung tumours. So the logic here is if we give them anastrozole, aromatase won't form, therefore estrogen won't be made. Therefore, less lung tumours will be present. But we've also got this bit here. If we give them fulvestrant, this binds to estrogen receptors. Now, why would that be useful? Well, here, if we look at the top, in these tumours, binding of estrogen to cell surface receptors stimulates cell division. If fulvestrant binds to estrogen receptors on the tumour cells, cell division can't be stimulated. So if we go with both, we're protecting it from both angles. So we're using the information provided to suggest why they predicted this. So straight away we can say, well, anastrozole reduces prevents, reduces estrogen production. We know that there because it inhibits the enzyme which will prevent, reduce estrogen production. So I'm just going to put A for anastrozole. So A prevents estrogen production. And then fulvestrant stops any remaining. So then fulvestrant would stop any remaining estrogen being able to bind to the tumours, meaning estrogen from binding to the receptors. So we know that anastrozole isn't necessarily going to work 100%. It's going to reduce the amount of estrogen. If we just get fulvestrant on its own, it's going to go onto those receptors. It's going to prevent the estrogen from binding. But is it going to work over time? Is it going to be some estrogen that probably can get in and bind some of those receptors? Yeah. Similarly, if we just gave anastrozole, there'll probably be some estrogen that would still be made and could still get attached to those receptors. If we use both, we're sort of blocking both channels, which is the main idea. So you'd get one mark for basically saying how the anastrozole would work, and then another for saying the fulvestrant, and then that would give you the 2 marks. Question continues. We're now given the data. So at week 15, the lungs of the mice were removed, and then they examined them, and the scientists then determined the number of tumours that are present and the mean tumour area for each group. So we get given this data here. We've got 2 graphs. This is going to be really annoying. So we're going to have to go back and forth. So I'm going to go back to that in a second. We'll look at the question first. So the question is, the scientists conclude that both drugs should be used together to reduce the risk of lung cancer in women exposed to tobacco products. Do you agree? Explain your answer. 5 marks. So what you need to do is basically come out with a yes or a no to start off with. Do you agree? Well, let's have a look. We need to remember what group Q, R, and S is. So group Q is our control. Group R is just anastrozole. Remember, anastrozole, that one stops the enzyme being made, and fulvestrant stops the estrogen binding. I'm just going to put bind. And then group T is both. So if we look at control, we've got our number of mice, number of tumours in each group. So on a control group, nothing's happened to them. So 4 mice had 2 tumours, 1 mouse had 4, 2 mice had 6, 2 mice had 7, 1 mouse had 8. Group R, which is the anastrozole, which is stopping the estrogen. We did have some with only with none. We had 1 with 1, 1 with 2. We still had 3 with 3, 3 with 4, 1 with 5, but we didn't have any up in the 6, 7, 8. So it has reduced how many each individual mouse had. But then groups S, so the size of them, again, we don't have them super large ones in 6, 7, 8 or 5, but actually we have a lot of them in 6, which is stopping that estrogen binding. But if we look at group T, we've got 5 with 0 and 5 at 1. Look at next graph. Key mean results. So the mean tumour area here. Control group, the mean tumour area is about 2.5. On anastrozole, again, we're looking at about, I know, so that one was probably 2.6, 2.5, just with S, 2, 4, 6, 2.6. But with T, we're looking at 2. So we've got a large difference there. So do we agree that we should use both of them together? Well, the first part of this answer, I'm just going to jot the answers down here and sort of work it through here, would be yes. We'd want to say yes, that you should use both together. From the data that you've been given, you should be able to conclude that. So let's have a look. What do we want to say about this? let's look at this graph first. Well, when we use them both together, we have less tumours per animal. So when in group T, we should use that one because it has the least amount of tumours per animal. And I would like you to often say, if we've been given something like figure 1, say as seen in figure 1, or just fig 1. Let's have a look at figure 2. What can we get from that? Well, we did that quick summary. It has that lowest mean tumour area size. So it's got the lowest mean tumour area size, and that's from figure 2. So straight away, you've got 1 mark, 1 mark, 2 marks. It's a 5-mark question. What else can we say that's still in a positive? Now we need to remember the question asks you to explain your answer. So we're at A level, we're going to maybe need some for and against because it's not all going to be strongly for it. Let's have a quick look. When we look at the top of ranges on our range of results for whisker plots, it has the lowest top of range as well. So even with the lowest mean, it still has the lowest top of range as well. So that's great. So we could say it's got the lowest top of range in tumour size. That would be figure 2 as well. But let's look at these mean tumours. Let's just zoom this in a minute. Oops. I don't want to draw all over it. I'm just drawing up with this now. This is not helpful. Here we go. Just going to get rid of all that random drawing I've done. Let's have a quick look. These mean tumours are all, it would help if my handwriting was a bit smoother. It's there. Within 2 and, where's that one? 2.6. So they're all within 0.6 of each other, the means. So we don't actually have that great variability here. So that is something that you could say. So you can say, yeah, you should use that one because X, Y, well, yeah, should use both of them together because when you use both together, we've least amount of tumours per animal, as we can see in figure 1. We've got the lowest mean tumour area, as we can see in figure 2. We've got the lowest, oh, that's another mark, lowest top of range in tumour size, as seen in figure 2. However, and this is where you're going to get them extra marks now. However, the mean tumour size, mean tumour area, sorry. So our mean tumour areas are similar. That will get you a mark just by saying that they're similar. We haven't got a large range in them. So let's have a look at them in a bit more detail. Let's think about them in detail. Actually, our ranges are overlapping. So if we look at the range of results between Q, R, S, and T, the ranges overlap. So that isn't great. So they share values. So we've got a sharing of values, which means differences might not be real. Means differences might not be real. So that would give you another mark. So that would give you 5 marks. A couple of other things that you could say, and I'm just going to talk through them with you now, even though we've got to the typical 5 marks, is this. We could say that the range might be affected by outliers. Standard deviation usage would be better. we've only done this on mice, so we haven't done this on any other animals. And in here, we're starting to talk about using it in women, but actually the experiment has only been done, only carried out on mice. So you might want to pick another mammal or even humans to try this out on. Also, the sample size, it was only 10 mice. We haven't done this on many mice. Could have side effects. We don't know what the side effects are. So side effects, question mark, what could they be? And we only did this for 15 weeks. So the time constraints of this. So there are 1, 2, 3, 4 more extra potential marks that you could get, and also talk about standard deviation from them. What I've really focused on there is the data ones to look at, but actually they're all just as valid sort of but parts to them. So what then happens in the question is it says scientists use tumour area as an indicator of tumour size. Explain why tumour area may not be the best indicator of tumour size and suggest a more reliable measurement. Now, suggest is quite good. It's saying that you can just suggest anything as long as it's reasonable. It could be a valid answer. So cells, as we know, are not flat 2D structures. Cells have depth to them. So just because a tumour might be wide, their area might be big, that might not actually be really very indicative of its size, because it could be very deep or it could be very shallow. So you could have one tumour that's got a very, very large area but very shallow, another tumour that's got got a smaller area but it's a lot more deeper. So actually in reality it's actually larger. So when we're talking about tumour size, we need to actually take into account depth, because tumours are 3D, not 2D. So something along the lines of tumours may have different depths. Tumours have different depths, and these need to be taken into account. They are not 2D. And then suggest a more reliable measure, measuring either tumour volume, mass or weight. So I'm just going to pop all 3 of those options. Yeah, you can say mass, volume or weight. Anything really. I mean, at this point, we've already removed the lungs to look at the tumours. We may as well get the mass, so we actually know well, how much physical tumour is there. So part E, it is a very long question, and I wanted to pick this one out because this is one where it actually uses lots of information. So the scientists repeat the investigation. This time they don't give the drugs until week 9. Suggest, suggest why they give the drugs at week 9 rather than week 5. So if we go back, and this is something I'd always say in questions like this, you've answered so much you can't really remember probably what's going on, because I can tell you now I can't remember what's going on the question. Here we go. Scientists investigate whether 2 drugs could prevent tumours in female mice. First, they remove the ovaries, then they inject the mice with a tumour-causing chemical found in tobacco twice a day, and the mice were then randomly allocated, and then they're given the drugs from week 5. So why might we leave it to week 9? Well, it could all to do with tumour growth, really. It allows tumours to grow, develop, form. We're giving the tumours longer to grow and develop. So allows tumours time to grow. It may be that actually just those 4 weeks of giving them the injections of the carcinogens that can cause the tumours, then you've only left it a week for those tumours to really grow. Reality is we might actually want to leave it longer to see how they grow. So that would actually help us investigate treatment rather than prevention, because actually in that situation we're probably looking more at the prevention if we do it straight after when the drugs come into the body. And often in humans we're not treating the prevention of cancers in somebody who's got carcinogens in the body. We're treating a cancer that's been found. So this allows the study of advanced cancer treatment. Oops, what have I just clicked on? Right, let me click on sharing this properly again. Yep. Advanced cancer treatment. So another group of scientists is currently using these drugs in human trials. However, the control group is not being given a placebo. Suggest why placebo is not given and what is best to give this group instead. Well, in their placebo was literally nothing. It was neither drug. It's unethical. It could be classed as unethical, and so it's ethical. It's ethical issues here. So we could say it's unethical to give somebody who's ill no medication, because it's unethical, as that person is likely to become more ill by giving them placebo, more ill by giving them a placebo. What could we use instead? Well, we could use cancer drugs that already exist. So use existing treatment. So we could use other treatments, other drugs to treat that person. And actually that's quite good as well, because then we're comparing the efficacy, how well those drugs work, to drugs that are already there. Because what we can sometimes find is that whilst we can find these new drugs and go, oh yeah, these new drugs are great, these new drugs treat the cancer, they might not be any better than the other one, or they might be a bit better but they cost more, they might have worse side effects. So this actually allows us to actually do some extra further analysis into it as well. So that's that first really super long question. Now we've got some more basic generic cancer questions. So a mutation of a tumor suppressor gene can result in the formation of a tumor. Explain how. This question, you want to pick out things like mutation, tumor suppressor gene, and it can result in the formation of a tumor, and explain how. So we know that if a tumor suppressor gene is mutated, it becomes activated. So I'm going to call it a TSG for tumor suppressor gene. So if a TSG has a mutation, it can become inactivated. That's your first mark. So if tumor suppressor gene has a mutation, it can become activated. This can cause the rate of cell division to become too fast. I'm going to point out where the marks are in here. TSG has a mutation, it can become inactivated, can cause the rate of cell division to become too fast. In this, you could say instead the tumor suppressor gene is not able to control cell division, it's not able to slow the cell division, but anything about it becoming inactivated, and thus cell division is then out of control and it occurs too quickly. Then we're asked, not all mutations result in a change in amino acid sequence of the encoded polypeptide. Why? This is because the genetic code is degenerate, and that is all you have to say. Oops, explain why. The answer is the genetic code. Genetic code is degenerate. And remember, what we mean by that is that more than one triplet code codes for one amino acid. So the next part of this question, some cells have a receptor protein, some cancer cells, sorry, so let's highlight that, cancer cells, we'll talk about cancer cells, have a receptor protein in their cell surface membrane, and that binds to a hormone called growth factor. This stimulates the cancer cells to divide. Scientists have produced a monoclonal antibody that stops this stimulation. Use your knowledge of monoclonal antibodies to suggest how this antibody stops the growth of tumour. At the minute, yeah, I'll have a little quick, yeah, we'll have a little talk about that. Actually, how many marks was that question? One was just one. We haven't redone monoclonal antibodies since you'll have covered them at GCSE. But let's have a little quick chat about them. So let's open up the whiteboard, actually. Let's have a quick chat about antibodies. Microsoft Whiteboard. So let's have a quick look at monoclonal antibodies. Oops. Here we go. Monoclonal antibodies. So monoclonal antibodies. Basically, how antibodies work is they bind to antigens. So, and antigens are found on viruses. So antibodies, they bind to antigens. Now, it's not just anybody binds to any antigen. It's very similar because it's a protein to how enzymes work. Our antibodies, oops, that zoomed out a lot. Our antibodies have, and because it's protein, tertiary, a specific tertiary structure. Or you could say a specific binding site, or you might have heard it referred to as a variable region. Any of those three terms are fine. You'll see that in textbooks, all three will get interused, but you are fine using any of them, either a tertiary structure, binding site, or variable region. So they have a specific one, and that is one that is complementary to the specific, not binding site, why have I wrote binding site? Specific to the receptor proteins. So let's have a quick look at that question. So use your knowledge of monoclonal antibodies to suggest how this antibody stops growth of tumours. Some cancer cells have a receptor protein in their cell surface membrane. For growth factor, this stimulates cells to divide. Monoclonal antibodies stop stimulation. So that information I just told you about monoclonal antibodies, them having that specific shape, will get you your first mark. So you're just basically going to explain about monoclonal antibodies having that specific tertiary structure. So I'm going to pop here monoclonal antibodies have, my screen has gone. Monoclonal antibodies have a specific tertiary structure. And that tertiary structure, so that'll be 1 mark, is complementary to the receptor proteins. Because it's complementary, it binds to it, and when it binds to it, it then prevents growth factor binding. So basically, they're your 3 key points. You just need to suggest that monoclonal antibodies have a specific tertiary structure, which is complementary to receptor proteins. If a monoclonal body binds to that complementary receptor, binds to the receptor protein, it will then block and prevent growth factor, which is our hormone that we're talking about here, being able to bind to those receptor proteins. Just had a moment, I can't remember what word for them. So methylation of tumor suppressor genes can lead to cancer. So let's have a quick recap of what methylation is. Methylation is that addition of a methyl group to our DNA. And it's really important in DNA because we use methylation for regulating gene expression. So that DNA methylation controls whether or not a gene is transcribed and translated. So we can have hypermethylation or hypomethylation. So tumors can occur from abnormal methylation of certain cancer-related genes. So in tumor suppressor genes, for example, if they become hypermethylated, and that's PER, of course, hyper, and PER, HYPER, not hypo, the genes cannot be transcribed. So proteins slow cell division. So explain how methylation of tumor suppressor genes can lead to cancer. So hypermethylation of the genes, so HYPER, I just need to, basically, I just keep spelling it because it's easier to explain. I'm saying hyper than hypo. So yeah, so hypermethylation of a tumor suppressor gene, TSG, prevents transcription. That will get you 1 mark. You're stating what it does. That hypermethylation of the tumor suppressor gene prevents transcription. If transcription, so then, but basically we're looking at how it leads to cancer specifically. So if transcription is prevented, so basically now we're basically going to explain what a tumor suppressor gene does. So if we methylate the tumor suppressor gene, it won't be transcribed. It won't go through transcription. So if we hypermethylate it, it won't be transcribed. Therefore, the protein produced from the tumor suppressor gene that prevents cell division is not produced. Second mark. The hypermethylation of tumor suppressor genes prevents transcription. The protein produced from a tumor suppressor gene that prevents cell division is not produced. Therefore, there is no control of mitosis. No control of mitosis, which can lead to the formation of cancer. And that will get you your 3rd mark. So basically, the question is basically wanting you to say, what happens when tumor suppressor genes are hypermethylated? Then we've got to link that to it leading to cancer. Well, basically, what is a tumor suppressor gene then? Okay, it makes a protein that prevents cell division. If we're not going to produce a protein that prevents cell division, that we've got no control of mitosis, which then will lead to potential formation of cancer. Okay, so here we've got another sort of data question. There's lots of data questions in the cancer section. So scientists investigate a possible relationship between the percentage of fat in diet and the death rate from breast cancer in women from 10 countries. Their data is shown in the table below. So we have percentage of fat in diet of the population and the death rate of women from breast cancer. The question is, describe how you'd plot a suitable graph of this data and explain your choice of graph. So we've got one variable against another, okay? Your suitable type of graph for this is we've got percentage of fat against death rate. We would want to do a scatter graph for this. A scatter graph would be sensible. So first mark, say what type of graph? Scatter graph. 1 mark. If you don't say anything else than that, you only get that 1 mark, but we need to explain your choice and how we would plot it. So what would that scatter graph look like? Well, basically, you're going to want to say what's going to be on your x-axis and what's going to be on your y. On your x, you're going to want to put percentage of fat in the population. Your y-axis, you're going to want to put the death rate. So here, we just put on the x-axis, you would plot fat, y, plot death rate. That will give you a second mark. Third mark, why? Why are we using a scatter graph? Well, we're using a scatter graph because we're looking at a relationship between two variables. And it's not just two variables, it's between two discrete variables. Okay, they're two independent variables. And we're looking at them and we're looking at the relationship between these two separate variables. What can we conclude from this data? Well, let's have a quick look. So that's obviously fat, that's death rate. So quick conclusion from that, apart from some quite obvious random outliers, there's a general pattern is as fat increases, so just death rate, we could sort of pop out some outliers. You could argue, you could either say this is an outlier and this is an outlier, or we could say this is an outlier and this is an outlier, but that's our general trend. So our general trend is our trend would show a positive correlation between fat in diet and death rate. But we have a number of anomalies. So we would just put here, but there is a number of anomalies. It's only worth 2 marks number. And we'll just have a look. No, we're not going to look at that one. That one's quite a long, complicated one to finish off on. Oh, okay, we will do that one then because they're pretty much at the end of these. Okay, we'll do this one then. Okay, so we've got metastatic melanoma. It's a type of skin cancer. So metastatic melanoma, type of skin cancer. It's caused by a faulty receptor protein in the cell surface membrane, and there have been no effective treatments for this cancer. So we have a drug, dacarbazine is a drug that's used to treat it because it appears to increase survival time for some people. Doctors investigate the use of a new drug to treat metastatic melanoma. They compare the median survival time for 2 groups. So we've got a control group who've been treated with dacarbazine, which is the old drug, and a group of patients who've been treated with dacarbazine and the new drug. The survival time is how long a patient lives after diagnosis, and the doctors also recorded a percentage of patients showing a significant reduction in tumours with each treatment. The total number of patients in the investigation was 502. and the table below shows the doctor's results. So the doctors compared median survival times for patients in each group. Oh, a bit of maths. How would you find the median survival time for a group of patients? It's a nice bit of maths here. We're going to rank all our survival times in ascending order. Thinking back to basic maths here. So we would rank survival times in ascending order. So obviously remembering the median is just that middle number. And we would find the value with the same number above and below. You can just say find the middle. So in many trials of new drugs, a control group of patients is given a placebo that does not contain any drug. Control group in this investigation has been treated by dicarbazine. Suggest why, like we said before, not ethical to fail to treat cancer. So it's just that ethics part again. We can't not give someone a drug if they've got something like cancer. So journalists who read this investigation concluded that ipilimumab improve the treatment of MM. Does the data in the table support this, and give a reason why? So median survival time is higher, and we had a high percentage of patients showing significant reduction in tumours. So we could say yes. How many marks is this? 4. So it's 4 marks. If your answer only relates to yes or only to no, you will get capped at 2 marks. So yes, those 2 things I've just said. So median survival time. How much is it increased by? Median survival time is increased by 2.1 months. And then percentage has changed from 10.3 to 15.2. Our percentage of tumour reduction patients increases. However, this is where you're going to get your other 2 marks from. No. We've used the median. The problem with the median is we don't know what our standard errors are. There's no statistical tests carried out. We don't know if this is due to chance. We sort of don't really know. So there's a lot of different variables here. So anything along the lines of these potentials. So you could base the fact that there's been that the data isn't reliable enough because standard error hasn't been used. Or stat test. I'm just going to put stat test here, something like a t-test. So cannot tell if the differences are statistically significant. Fab. Right, I'm going to stop there for today. We'll do some chemistry tomorrow, and I will see you at